Hi, everyone, and welcome back to the channel. So today I'm going to be talking about genomics. And more broadly, this particular overview that I'm going to give could really be classified as the omics topic. If you follow science, particularly the biological sciences, and you've been doing so for a little while, you may have heard people talk about the fact that in many ways we are in the era of big data, which means we now have these giant data sets that we get about whatever topic of interest we have in the biological sciences. And we now need people to be able to go through and sort of parse this data and find out what it's actually telling us. So I'm going to have some videos coming out in the near future where I go into more detail on some of these different techniques. Not all of them. There are far too many. I would have to do an entirely different channel to just go through all of these techniques in great detail. But what I do want to do in this episode is give you an idea of the kind of information that we can find out, give you an idea of some of the techniques that are used to find this data, and kind of give you an idea of why this stuff is useful. It maybe help you understand when people are talking about being in the era of big data, what we're actually talking about. So before we get into some of the modern stuff, let's back up a quick second and talk about what old school genomics looked like. And to call it genomics, really is really a bit farcical, simply because this was done on such a small scale that it really wasn't omic-y enough. It wasn't big enough to be considered an omic. But let's talk about it. So when we talk about genomic analysis, we're really talking about how can we learn more about what the genome, what the sum total of genetic information of an organism actually looked like. So original genomic analyses relied on a few things. First, spontaneous mutants. So if you were studying fruit flies, or you were studying C. elegans, or you were studying even mice or something like that, and you happen to notice, in some population, hey, there's a really weird variant here. There's some mutant phenotype that I'm seeing. Either it's got too many legs, or the wings are curled, or, its eyes are a different color, or it has too many eyes. That would be a spontaneous mutant. You didn't do anything to cause that. It just appeared. And the earliest investigations into, understanding the genetics of fruit flies, for example, were based on that. Hey, look, there's something wrong with this one. Let's see if we can figure out what makes it different. As we got more and more into studying these things and we learned more about organisms, we also learned ways that we can induce mutations. So, for example, maybe you found this one weird dude in your tube of fruit flies with curly wings. And then you went ahead and said, okay, well, what if I expose them to some type of chemical mutagen, or did something to sort of induce mutations, and see if I can get more flies with the same phenotype? Then I could analyze those and try to figure out if I could identify, what the gene is that's responsible, or at least, where it's located. Now, in a previous video of mine, I talk about how a lot of the mapping of genomes early on was done through linkage analysis. So you might take, your curly winged mutant and your weird eye mutant and say, okay, well, these are probably controlled by two different genes. They're two entirely different phenotypes. Well, let's find out if we can find out if they're on the same chromosome. Are they syntenic? Are they linked, or whatever? Okay. And a lot of research in the first half to the middle half of the 20th century was done doing just that. They were using techniques like these to map the chromosomes in fruit flies and mice and bacteria, worms, yeast, viruses, you name it. The problem is this is incredibly time consuming. You've got to set up matings. You've got to set up a lot of matings because you've got to get, decent numbers so that you can calculate the recombination frequency and all that sort of stuff. And by the way, if these alleles had no phenotype, so if they were unapparent, or if the phenotype for a mutation was lethality, completely useless. There's nothing you're going to be able to do. How are you going to follow the phenotype? How are you going to, determine where, if they're linked or not, if the progeny die, if they get the mutation, right? Even more of a problem is a lot of this understanding was helping us sort of set the groundwork for, okay, great. but I only care about worms so much. I only care about fruit flies so much. I'm using these organisms as models to help us better understand the genomics of human beings. Well, again, you can't set up linkage analyses of humans. There are some pretty profound ethical concerns that come out of this. So instead, we had to rely on spontaneous or inherited mutations in humans, and do things like pedigree analyses in this. And there's really only so much that we can, learn through these types of things. So if we fast forward a bit into the 1970s, we end up with a technique known as Sanger sequencing. It is named after the PI of the lab who discovered it. But Sanger sequencing relies on radioactivity. It relies on gel electrophoresis. It's a very cumbersome technique, but for the first time, there was the capability to sequence, to actually learn the A's, C's, G's, and T's that are found in a gene. So compared to modern techniques, which, we can get tens or hundreds of millions of bases uncovered in a matter of hours. I mean, you could get, one of my graduate advisors basically said, like, in the 80s, all you had to do was sequence the gene and that got you your PhD. Like, that's how involved it was to be able to do this, which is great at the time. But for now, in modern era, it's just far too slow. It's too low throughput for anything to be able to do anything with it. But nevertheless, we have to look at Sanger sequencing as sort of the beginning of the genomics revolution, because for the first time, we were able to go through and map at least a single gene and figure out what A's, C's, G's, and T's were in it. And then we could figure out if you have a mutant, we could sequence the same gene and see what is different between the mutated version and what we would now call the wild type version. All right. By the time we get to the 1980s, we can start using restriction fragment length polymorphism, so RFLPs, basically taking small chunks of a chromosome, sequencing those chunks, and then sort of stitching those chunks together based on sequence similarity to show, like, this is supposed to be after this, after this, after this, right? It's a bit like putting together a DNA puzzle, which means we could now map, parts of or entire chromosomes at this point. All right. We could then use things like pedigree analyses to link genes to genetic diseases. We call this, there's a thing called positional cloning that I won't get too far into. But basically, what that means is we were able to start sequencing significant chunks of an organism's genome, but it needed to be done sort of one at a time. And by the time we kind of got to the end of this stage, the end of sort of Sanger sequencing's useful lifespan, we had gone through about 3,500 human genes, which is about 10% of the human genome. By the time we get to the 1990s, computer processing power had improved significantly. And rather than quite literally having people sit together with, long strips of paper and just sort of matching them up by eye, we were now able to harness the power of computers to do this for us. And this, for the first time, made it possible to be able to sequence the entirety of complex eukaryotic genomes, including the human genome. All right. The approach to doing this is usually referred to as shotgun sequencing. So you would take an organism's genome, use these enzymes called restriction enzymes, which cut DNA at specific sequences. I'm giving you some examples of these down here. And then you would take these small chunks of DNA, you would sequence them, and then basically what you would do is just use the computer to stitch the whole genome together, essentially, by looking at, okay, these 4 overlap, so this must be the next stretch. Then there's this overlap, that's the next stretch, and then you can say this is what the assembled sequence looked like. Again, it's sort of the same puzzle that humans were doing in the 70s and 80s, but by the time we get to the 90s, we now have computer processors that are capable of sort of doing this for us, so we don't have to. Now, what you need to understand is, again, 1990s computers, I'm old enough to remember those. The amount of processing power in a 1990s computer is significantly less than what is in your cell phone right now. So, picture your cell phone trying to assemble the human genome, and now you understand what scientists were using at that point. But nevertheless, by the end of the 90s, by the early 2000s, we were able to begin really in earnest sequencing entire genomes at that point. And this is kind of describing the process. Here's an example of what you might get, right? You sequence a genome, you get this, the computer overlays it and says, this is now your reference sequence. But the other thing is now you've got to go through, you can't do it just from one person because everybody's unique. I would say in general, on average, every person has about 100 different mutations that even their parents don't have. So even in the most similar of circumstances, there are differences. So now what you would do is you would sequence multiple individuals within a species, overlay those and be like, okay, well, this, basically like the majority of people have a C here. If this person has a T, that's a single nucleotide polymorphism that sets them apart from the whole population, right? Or, most people have a GA here. This person has a deletion here because they don't have that GA or whatever. All right. So this is kind of how we do that. And then what we do is we take the large data sets and say, well, most people have a C here. The reference sequence should have a C. If somebody doesn't have a C, that's a polymorphism. Most people have a GA here. If they're missing one or two of those nucleotides, that's on their end. It's not representative of the individual as a whole. And by the time we get to the 21st century, by the time we get to the year 2000, we now have the ability to generate something like 2 million bases of sequence every 24 hours, which again, we have to set into the context of the human genome is about 6.2 billion nucleotides. So you're still looking at a few months before you can sequence the whole genome. Nowadays, this presentation is slightly older. I said new techniques are about 50,000 times faster. It's probably significantly more than that because every year our processing power gets faster and faster. So like that's now. If you're watching this, a year or two from when I've created this, it's more. All right. Which is great if you're into sequencing genomes. So once we get a genome mapped to a large extent, we publish something called a draft genome. This goes for all organisms, not just human ones. Anybody that publishes a draft genome is very well aware of the fact that there are probably some holes in the sequence. You'll just see like a gap there that isn't there. More work needs to fill those gaps. But sometimes there are regions, I won't get into the details, there are some regions of genomes that are particularly tricky to sequence, especially if they're highly repetitive. You get errors where some of the repeats don't show up. And there's a question of, how many times does this sequence repeat? Or is it supposed to be 9 adenines in a row? Or is it only 7? That kind of stuff. Once you've done this over and over again, researchers will be happy that they've, once they're satisfied, we've minimized the amount of error that's there. They will now refer to that as a reference genome. And then at that point, what we have in the reference genome is considered to be the wild type. And any time you see a deviation from the reference genome, it's generally considered to be a mutation. Although you should never consider a reference genome to be 100% accurate because nothing's perfect. We're talking about analyzing billions of nucleotides in some cases, and it's just not possible to get a perfect readout of that. Once we have reference genomes, once we started having piles of reference genomes, so like now we have compiled so much genomic information. We now start to talk about the field of bioinformatics. So when we talk about bioinformatics, we're talking about the use of computers and mathematics to organize, share, and analyze data. That data is typically going to refer to things like gene structure. We'll be talking about genetic sequences. We can talk about expression patterns. We can talk about protein structure and function. And one of the things that's happened really in the last 20 years is what we call the omics revolution, where there are now, genomics, proteomics, viromics, that kind of stuff, looking at all this type of stuff. And common research projects within this field of study They're comparing sequences. They're trying to identify the location of genes, the promoter region of genes, the enhancer sequences. They try to identify where transcription factors bind so they can get an idea of which transcription factors may regulate certain genes, because then we can begin to understand which signaling pathways lie downstream of controlling these genomic regions. We can look at certain chromosomal regions that may be more important than others. We can predict protein sequences, protein structure, protein function. And if you're in the field of evolutionary biology, these are hugely important because now we can use genetic information to uncover evolutionary relationships. And we find some very surprising things when we've done that over the last 25 to 30 years. Now, most of the research that goes into doing to going through and establishing reference genomes, to doing these, are funded, at least in the United States, through public funding, which means they need that data needs to be public information. And as such, there's something called NCBI GenBank. This is a warehouse for publicly available gene sequences. Over 200 billion stored sequences. It's far beyond that. But again, you're understanding, like I said, it doubles every 18 months. So, if I made this, if it's a year and a half from now, we're at, 400 billion, the number of the amount of bioinformatic data available is just astronomical. It's overwhelming. All right. And there are tools that go along with this, basic local alignment search tools called NCBI Blast. You can literally put a DNA sequence into this and it will find similarity between any organism that's in the system. So it's insane the amount of information that's out there about genomic sequences. And it's so helpful for evolutionary biologists, for people interested in life sciences to be able to find these things, because you can compare and identify and find similar sequences between different organisms. If you're interested in studying C. elegans and you want to study, a human disease, you use this to say like, okay, well, these are the human genes that are mutated that cause this disease. What genes should I be looking at in fruit flies, in worms, in mice, in monkeys, in dogs, in cats? You pick an animal, you pick an organism, and you can find it. You can find out what's similar between a daisy and, a cat in a cow, based on this sort of stuff. So really, really interesting, powerful, awesome things that are out there. And the cool part is completely freely available since this was paid for by tax dollars. So let's talk about some fields of study within bioinformatics. One of those is called functional genomics. So functional genomics is interesting because the goal of this really is to use sequence data to predict gene and protein function. So how do we do this? Well, what you do is, if you are interested, like I said before, if you're studying one organism, okay, and you're interested in studying something that happens in humans using this organism, you would be curious to say, okay, what is the homologue to this gene in humans in C. elegans, okay, which is a nematode that's frequently used for genetic research. Now, I want you, because I'm going to discuss some of these things, I want you to remember some terms that I've talked about in previous videos regarding homologous structures and analogous structures. So if you are looking for something that is a gene that is evolutionarily related to a gene in another organism, that is considered a homologous gene. So a gene homologue is this gene is the ancestor of this gene, but in a different species. Does that make sense? It's like a homologous structure. It's evidence of shared ancestry. This is typically what we're concerned with, because if you want to study something in rats, mice, worms, fruit flies that has an impact in a human being, if you want to study a disease or something like that, you need to make sure you're working with the homologue. Okay, because that's what's going to give you the most similarity. There are also analogs. All right. Analogs are like analogous structures. They are genes that will lead to a protein that performs the same function, but is completely unrelated evolutionarily. Basically, this particular organism has a gene that evolved on its own to do the same thing. So again, this is sort of like bat wing and insect wing, right? So flies have wings that allow flight, but they're not related to the wings that allow flight in birds. Same thing with analogs. No evolutionary relationship, but does the same thing. And then we get into two topics that we really haven't talked, two sort of terms that I don't have a sort of a direct relationship to my conversation about evolutionary biology: paralogs and orthologs. Okay. So a paralog is a homologous gene within the same species. For example, humans have an alpha and a beta hemoglobin. We have two different genes, alpha globin and beta globin. All right. They are clearly homologous to each other. Okay. Why? Because they're identical in many ways. It's very clear that they at one point were a single gene. And when we see paralogs in an individual species, these are likely the result of gene duplication. So at some point, there was a single hemoglobin gene. There was a gene duplication event. And then we end up with two genes that perform similar but different functions: alpha globin and beta globin. And in humans, to get a functional hemoglobin protein, you need two alpha and two beta globin proteins. Okay, these are paralogs, homologous genes within the same species. On the other hand, orthologs are homologous genes between different species. All right. So if I look at the beta globin gene in mice and the beta globin gene in humans, those are considered orthologs. All right. So paralogs and orthologs are subcategories of homologous genes. Analogs or analogous genes have no evolutionary relationship. So really, you could think of these two here as being subgroups of homologous genes. Are we talking about homologous genes that exist within the same species, paralogs, or are we talking about homologous genes that reside in different species, orthologs? Okay. Now, another aspect of functional genomics is trying to analyze protein function based on domain analysis. Okay. So what that means is the old school way of looking at protein structure is something called X-ray crystallography. It's a very complex process, but it does allow researchers to discover what the basic domain structure of various proteins looks like. It's super powerful, and it's really the only way we have of knowing exactly what a protein looks like, structurally speaking, because you can't see them with a microscope. You can't see them with the naked eye. So you actually rely on X-ray diffraction to get this done. Again, super complex. We don't need to get down this rabbit hole, but what we can do with functional genomics is, because we have a lot of this information already, and because we have a lot of genomic information about proteins whose structure and function we know, we can then begin using that information to predict what parts of a gene will encode what parts of a protein. Now, again, this is a little bit of guessing. I don't like the term guessing so much just because it implies that we're just like, I don't know, maybe. It's not. It's informed guessing. It's a hypothesis, really, is what it comes down to. This is a way of developing a hypothesis about what parts of a protein will serve which function based on what we know about proteins who we've definitively identified. So, for example, we can take a protein whose structure we know nothing about. We can look at the gene. We can look at the amino acids encoded by that gene, just simply because we understand the genetic code. And we can begin identifying which sequences within the protein will likely play which function. So, for example, which part will form the channel for the ions to go through? Which part will be membrane spanning domains? Which part could be a DNA binding domain? Which part could be where targeting sequences, like to get it to where it's supposed to go? Which parts of the protein act in that capacity? And we can do that because we have existing information. And functional genomics allows us to go through and provide some amount of predictive power to, like, okay, here's a new protein that I'm just starting to study. Let's look at it from the amino acid. Let's look at it from the nucleic acid perspective and say, can we predict that this is probably a transmembrane domain? Why? Well, there's huge stretches of hydrophobic amino acids that would make sense. We know this is a transporter. It's got to be in the membrane. These are probably the parts in the membrane, right? So some really interesting stuff that we can do there. Another way that we can analyze proteins is by looking to see, what exactly they do. So, for example, there are a group of proteins called transcription factors that we find in eukaryotes. I've talked about them in previous videos. There are these proteins whose job it is to sort of respond to signaling pathways and then decide which genes should be expressed or not expressed in response to the changing environmental conditions, right? So you get a change in the environment, the cell senses that through the activation of receptors or inactivation of receptors. Those receptors then send a signal through a signaling cascade, eventually reaching a transcription factor. And then the transcription factor goes to the DNA and interacts with it in a way that either encourages gene expression or silences gene expression. So if you were curious to know, like, okay, well, I'm very curious to know which genes are regulated by a given transcription factor, you could use a technique known as ChIP-seq, which stands for chromatin immunoprecipitation followed by sequencing. So what do? Well, you take your transcription factor of interest, you trigger whatever response you want that would trigger the activation of that transcription factor, and then essentially you take the cells, you lock it, you kill the cell while it's in that state. Theoretically, at that point, the transcription factor should be bound to the DNA. Okay. You then go through, shear up all the DNA. So anything not protected by the binding of that transcription factor gets destroyed, but the DNA that's trapped under the transcription factor stays bound to that transcription factor. You then use antibodies attached to these glass beads, usually, or plastic beads, to bind to the transcription factor. And then you get rid of the protein and precipitate the nucleic acids. So basically, you destroy all the DNA except for those protected by your transcription factor. You grab onto that transcription factor. You then release the DNA trapped by the transcription factor and then sequence the DNA. And then what you end up is getting a bunch of sequences. And these little sequences are those that are bound by the transcription factor, right? Well, then you take those sequences, you map them to the organism's entire genome, and you go, oh, cool. So this sequence belongs to the promoter for this gene, which means that under these conditions, this gene is being regulated by this transcription factor. And you can do that and find out that there are hundreds or thousands of genes that are being regulated by a given transcription factor in response to a given stimuli or a given stimulus, which is really powerful because now you're understanding, like, okay, when I do this, these genes get activated by this transcription factor. RNA-seq is similar in the fact that instead of looking at transcription factor specific binding, basically we're just looking to see which genes are expressed and which are not. Okay, and we do this actually by grabbing RNA. So if you recall, the central dogma of molecular biology is that DNA gets converted into mRNA through transcription, and then mRNA gets turned into a protein through the process of translation. Essentially, what we do here is we take a cell or an organism, whatever we're doing, whichever we're interested in studying. We isolate all of the RNA, okay? And then to make sure that the only RNA that we actually detect is that in the form of messenger RNA, we actually use reverse transcriptase, interestingly enough. We isolate the mRNA, we use reverse transcriptase, convert it back into DNA, and then we sequence that what's called cDNA, okay? And what you get is a relative abundance of how much cDNA is in that sample. What I mean by that is this. If you detect the messenger RNA of a given gene, that means that gene is being transcribed. So if you you simulate some type of condition, whether it's starvation or reproduction or whatever it is, you trap that cell in that state. And what you're basically saying is what genes are being expressed right now? What mRNAs can I detect? You convert it into cDNA because it's easier to do sequencing on cDNA than it is RNA. Okay. And you go, okay, well, at this precise moment, when I isolated the mRNA from this cell, here is the relative abundance of DNA, or relative abundance of mRNA that's being expressed. If you find a large amount of cDNA mapping to a certain gene, that tells you that that gene is being highly expressed under that given condition. If you don't detect the cDNA for a given gene, that means that gene is not being expressed under that specific condition. And again, this is very interesting if you're trying to build trying to understand how changing conditions impact a cell, what proteins are being made. And you can detect that by what mRNAs are present. The conversion to cDNA is just a technique to make it easier to do the sequencing. But again, we are getting very big data out of this, right? We're getting the expression of thousands of genes and being able to say, okay, well, when the organism is starved, these genes are in high abundance, these are in low abundance. Then if we look at an animal that's well fed, do they flip-flop? And the genes that change in response to that changing condition, those are interesting because it tells you those genes are being regulated in response to those changes. And if there is a disease or a phenotype or anything that is in a particular mutant at a given time, that can tell you which genes that you should be interested in. It might tell you which genes that are being tweaked. Or you might be curious to know, for example, like, is there a way that I can screw with that, right? Can I overfeed an animal but not get this change by messing around with a transcription factor or something like that? Again, we're trying to understand what's happening inside of cells, and this kind of data is invaluable to that. The last one I'll talk about is something called microarrays. Microarrays are really, really interesting. You basically take a patient sample, compare it to a control sample, and it literally goes onto these like silicon chips. And basically what ends up happening is you are able to detect using primer sequences and things like that, the relative abundance of gene expression. And you get these cool, really colorful maps where, like, if it's bright red, it's overrepresented. If it's dark blue, it's underrepresented. And if it's sort of like that coolish blue color, it depends on which array you're using. But if you get that sort of cool color, there's no change at all, which would argue that this gene is not regulated in response to these changes. But the key here is you got to have, like, this is more of a differential approach, if that makes sense. So what you're looking for is changes in gene expression under this. Okay. Again, microarrays, very powerful ways of getting large amounts of data and getting a big understanding of what is changing with respect to gene expression under varying conditions, or compared to two individuals who have, very different phenotypes. Somebody who's, prone to getting cancer, someone who's not, somebody who has a particular disease versus somebody who doesn't, that kind of stuff. So all of this big data and all of these techniques that started to get established around the turn of the century and have just only grown and become more refined over the last quarter of a century, actually, have spawned the omics revolution, where we started with genomics, sequencing of DNA. We now have people that are studying proteomics, which is doing very similar analyses, but of proteins within the cell. Metabolomics. This is analyzing proteins and enzymatic pathways that are involved in metabolism. So how does somebody with diabetes differ from someone who doesn't? How does somebody who's obese differ from somebody who isn't? How do certain diets impact people, right? This is really powerful information. Glycomics. So carbohydrates play a very important role in cell signaling and in cell structure, but they're also involved in our metabolism. So can we learn more about the expression or the content of carbohydrates within a cell? Toxicogenomics is really interesting. This would look at the effects of chemicals on genes, including, does it increase mutation rates? Does it alter gene expression following exposure? This would be stuff that would be very important in examining, for example, there's been a conversation in our country about, military veterans who were exposed to burn pits. Is this a problem? People who were exposed to Agent Orange during Vietnam. Did this alter the expression of genes? Did this cause mutations? That kind of stuff. Metagenomics. So this is analyzing the genomes of organisms that are collected from the environment, right? So rather than like, we're just studying this lab organism, can we grab like populations of this species of fish here and compare it to the same species, but in a different location, or related species, and do that? And another one that's gotten very big recently is pharmacogenomics. So one of the things we're getting a better understanding of, we've always known this, right? Drugs have side effects. Pharmaceuticals have side effects, right? But some people aren't bothered by them. Some people are bothered by them severely, and some people get a mixed bag. Why? Well, it likely comes down to our differences, the subtle differences that make me, me, and you. We also know that drugs have different outcomes depending on the patients, So one drug that works great to treat this cancer in this person doesn't do, even though it's the same cancer in this person, doesn't do a good job. Why does this antibiotic work well for me, but it doesn't work well for my wife? Pharmacogenomics can give us a better understanding of how genetic differences may contribute to the efficacy or the potential for side effects of a given drug, which means, theoretically speaking, one of the things that might happen in the future is you go in, you get your genome sequenced, and based on our understanding, your doctor might go, oh, you have this unique variant of this gene, and what we find is this drug is more effective in people with this variant. So not only are we going to get a better patient outcome, we may, get to the point where we're basing what drugs we give people based on this, right? Like, well, hold on, rather than checking your chart, I also have to check your pharmacogenome to make sure I'm prescribing the right antibiotic, the right blood pressure medication, the right chemotherapy for your cancer, which is really cool. And this is not even close to the full list. You name a field of study, and there's probably an omics associated with it at this point in time. Pharmacogenomics, like I said, is paving the way for genomic medicine. Like I said, drug is toxic but beneficial. Drug is toxic but not beneficial. Drug is not toxic but not beneficial. Same group, patient group, same diagnosis, same prescription, you get different effects. And the entire purpose of pharmacogenomics is to sort of build your own personalized library with your genome and say, like, maybe this drug will work better for you, so you don't have to cycle through 2 or 3 drugs before you find one that really works for you, which is incredibly powerful and super promising for the future of medicine. But we're getting there. That's all I can tell you. We're getting there. The last thing I'll end with, this is more something really cool from a biologist's perspective. It might not be something that everybody's interested in, but Stone Age genomics. So because we're getting better and better at these types of things, we're getting better at isolating DNA. We're able to use increasingly smaller amounts of DNA to get a genomic profile. We are able to now gather DNA samples from extinct species and compare those to modern species. That doesn't just include things like the woolly mammoth that I'm talking about here, or a horse bone. It also includes human ancestors and ancient humans. So this is allowing us to get a much better picture of our interrelatedness with Neanderthals and Homo erectus and other extinct human ancestors, and giving us a better picture of how human evolution may have occurred, as well as, interestingly, what parts of being human aren't actually coming from humans. So many of you might be aware that if you are of European descent, about 1 to 3% of your genome is Neanderthal. Like, it's not even you, it's Homo neanderthalensis. People of Asian descent, on the other hand, there have been at least 2 documented admixtures with an entirely different species of human called the Denisovans that we actually know very, very little about. And it appears, based on the genomics data that we have, that there were 2 different instances where these populations interbred into Homo sapiens. So again, just super cool. If you're into evolutionary biology, this stuff is just so cool. And we're learning day by day there's because we know that there's this sequence identity with African elephants and, the Siberian mammoths and stuff like that, we can get a better understanding of, can we bring the woolly mammoth back? And, there are people who are interested in doing that, saying, hey, the woolly mammoth coexisted with humans. We used to eat them. We hunted them to extinction. Should we just bring back the woolly mammoth? And because we're getting better and better with these techniques, we're getting a better understanding of how that might be possible, which I don't know. I guess we're in that conversation of we can, but should we? But it's cool that we're at least able to have that conversation now. So I hope you learned a lot. This is some really cool stuff. Like I said, in some future videos, I'll go into these techniques in a little more detail, but I hope you found this interesting. It is really cool and powerful information, and I can't wait to talk to you about it more in future videos. So thank you for tuning in, and I look forward to talking to you again soon.